research into infectious diseases is in the area of malaria research, and there's been some pretty amazing contributions from WEHI in that field, and Jake Baum certainly has contributed very significantly to the success of the division. And Jake's origins are actually, he studied at Oxford and then went on to the London School of Hygiene and Tropical Diseases, and we're very fortunate that he came over to WEHI, and he's really applied the sort of cutting edge technology of imaging to address many issues, fundamental issues of host-parasite interactions regarding malaria. So welcome, Jake, and thank you very much for giving us an overview of malaria and where we're going.
Okay, I was saying to Mark earlier, it might have been easier for you to just ask me to give an overview of life or biology, because to try and give you an overview of the whole of malaria, sort of virulence, resistant mechanisms of pathogenesis, is quite a hard task, but I'm going to give it my best shot. So I'm going to really try and introduce you to everything in malaria. Very little of it my own contribution or my own research. Some bits from research from the division. And if you leave with only one thing, then hopefully you'll leave with an appreciation for the complexity of the malaria parasite and its biology and our interaction with it. Okay, so most of you will be aware, whether it's your own generation or your parents' generation, of the 1950s campaign to eradicate malaria. It came on the back of DTT spraying and its success in killing vectors and draining swamps and clearing the land of this, this incredible pathogen, which has been with us sort of since the dawn of humankind. And in some ways, actually, the campaign was an incredible success, especially for areas of southern Europe, southern US, the Middle East. But most of you will probably be appreciative of the fact that the eradication campaign failed, and where it failed most dramatically was in the third world. So if you look at sort of these maps of distributions of malaria, it was basically a disease that covered the whole of the tropics and sort of southern areas of areas like Europe and the US. Post-eradication, it was a pretty amazing achievement in areas of like southern Europe. But where it really, actually, the resolutions the colors come out kind of strange. But the areas where the sort of net change has been so disastrous is areas like the poorest nations of the world, areas like Africa, India, South America, and they still have rampant malaria. So it's still an incredible burden on humanity. So, I mean, it's very easy to just throw out statistics. A child dies every 30 seconds from malaria. I've given a talk where I had a little ticker in the left-hand corner which clocked the number of children who should have died by the end of the talk. So, I mean, I know that everybody tries to get over the idea that their disease is the most important one, but hopefully you get an appreciation. This is a disease that affects hundreds of millions of people every year. So there's something like 10% of the world every year is affected by malaria. Something like a third of the world is exposed to the potential to get infected by malaria. And as many as a million deaths, although that's kind of argued about. But perhaps even more than the health burden is also the economic burden of malaria. Things like a third of all work or school absentees in some of these areas is a result of malaria. So we're dealing with a disease that is a massive economic force in the world. So I just missed one part there, which is that when we talk about malaria and the malaria parasites, we're talking about 4 or 5 different species. Plasmodium falciparum is the main one that people talk about when they're talking about death, but Plasmodium vivax is increasingly acknowledged as a source of both death and also disease morbidity. P. ovale and malaria I sort of thought to be mainly a benign kind of disease. Certainly the person gets sick, but it's not sort of dangerous. And P. knowlesi has just emerged as a new zoonosis in Southeast Asia, jumping from macaques into humans and actually causing quite lethality and some severe disease. Okay, so very brief overview of malaria parasite. It's in the phylum Apicomplexus. It's a eukaryote. In the third year micro course that I've given at Melbourne University, the number of students who put bacteria or virus in their essays is still shocking. So it's a eukaryote, it's got a nucleus. It's in the genus Plasmodium. It's an obligate intracellular parasite, so throughout its life cycle it must invade host cells in order to complete its development. 2 hosts, humans and mosquitoes, and the mosquito we would kind of classify as the definitive host because that's where sexual reproduction happens. And it's predominantly haploid through the genome. The main feature of that is that it means it's a real pain if you want to do genetic modification, because most of the time, being haploid, you have an essential gene, you can't just knock it out. So that creates a real problem for the genetics. Now, normally when you give a malaria parasite talk, this is the slide you put up and say, this is the life cycle, which I'm not going to talk about, and I'm just going to focus on one little bit. So what I'm going to do now is something I've always wanted to do, which is to gratuitously go through the life cycle, and really, hopefully it won't be too painful, and really just point out some of the beautiful aspects of parasite biology as we go through. Okay, so let's start off with the infectious bite. Actually, can I lower the lights? How do I do that? Lights. Dim too?
I'll come down.
You'll come too? Okay. Just so you get it. Here comes Norm. Talk amongst yourselves. Well. Sorry, Norm. Thanks. Just the front ones. That'd be great. Here we go. Okay. So we start off with an infectious bite. So what happens at the infectious bite? So this is a movie made of a mouse ear by Jerome Vanderberg's group at NYU. And what you can see here is a mosquito feeding into the ear, probing its proboscis in. And as it does so, it's got this challenge. It wants to get into the bloodstream to take a blood meal, but it must clot the blood. So stop the blood from clotting, so it doesn't actually jam up the proboscis. So as it does that, it injects saliva, and cunningly placed in its salivary glands are the salivary gland sporozoites, the parasites that actually establish infection, and they get released. This is another movie from the same group, and you can see the parasites spilling out of the proboscis into the surrounding tissue. So their parasite is now inside the mammalian host and has to basically get into the bloodstream. And this is the parasite, the liver stage, the salivary gland stage form, the sporozoite, a highly motile banana-shaped cell, about 10 microns long. Okay, so we've been delivered into the skin. Now we have to get to the main site of primary infection. It's still a silent stage infection. You don't know you've got malaria. This is hepatocyte liver stage infection. So here we have a parasite in the skin. This is incredible work from Robert Menard's group in Paris. The parasite circles a capillary, finds a way in, breaks through, and then gone into the bloodstream. And so it gets taken via the bloodstream all around the body, obviously finds its way to the liver, a very heavily serviced area by the bloodstream. And then they're recognizing, we think, sulfation levels on surface epithelial cells. The parasite then stops and then starts to cross the sinusoidal barrier and then basically establishes an infection in a hepatocyte. This is other intravital work from Ota Frevet's group. Okay, so we've now been in the liver for, depending on the species, approximately 2 to 14 days. There is then this incredible process of cell division. I think if you were interested in cell division and you wanted to pick a problem in biology to work on, this would be an incredible program of research to try and establish. Here we have one parasite goes in, so this is a primary hepatocyte being invaded by a sporozoite from Volker Hüssler's group, and then that single cell gives rise to tens of thousands of daughter parasites. Amazing achievement in sort of cell division, organelle segregation. And one of those sporozoite comes in, tens of thousands of these, what are called merozoites, come out. And then these are then released into the bloodstream as a bag called a merosome, which releases these infectious parasites into the bloodstream, which can then go on to establish the part of the parasite life cycle that we often think of in terms of disease, which is blood stage infection. Okay, so blood stage infection starts with merozoite invasion of the red blood cells. This is a movie a student of mine, David Rigler, took. So here we have the parasite. It's been released into the bloodstream. It meets a red blood cell. Oh, and then it's in. That's kind of real time. So it only takes like 30 seconds for it to get in. This is actually the only stage in the life cycle really where the immune system gets a good look at the parasite, other than, say, well, liver stage infection to some degree. But here the immune system in the bloodstream is really getting a good look at the parasite throughout its life cycle. The parasite then starts to develop. This is a recent movie from a paper from Tim Gilberger and Tobias Spillman's group, and you can really see the parasite dancing around. We have no idea what this means, but it's kind of funky. So the parasite sort of disco dancing inside the cell. I'll put it up there because if you've got a good idea what it is, then I think that'll be something really cool to work on. Then once it's in there, it replicates, and then bursts, and then releases, and then starts another round of infection. Generally, that takes, depending on the species, for Plasmodium falciparum, it's a 48-hour life cycle, so from entry to division to about 20 to 30 daughter parasites, about 48 hours, and this establishes the 48-hour periodicity of fever that we associate with malaria disease. Okay, so you've had now the blood stage life cycle. That basically continues, overwhelms the body, and you know, you get an incredible multiplication of parasite biomass in the body. One of the things that the parasite does so well is it manages to secrete proteins onto the surface of the red blood cell, which allows it to adhere to capillary beds. So this is called sequestration. So here, this is an in vitro experiment done by Brian Cook, where you can see that parasites are sticking to an artificial epithelia as the flow is massively going past. And so this enables them to sort of stop themselves from being cleared by the spleen by passing through the body. So they end up sequestering in deep vasculature. This was an experiment where they showed the actual ability of the parasite to withstand incredible shear force from flow and stick to an artificial epithelium. And then in these areas of sequestration, this could be in the brain, and you can already start to imagine what that would mean if you had parasite biomass sequestering in deep vascular tissue in the brain, or this could also be the placenta during pregnancy-associated malaria. And that can cause significant pathology, and in fact, the main cause of death is as a result of sequestration of parasites in those kind of deep vascular tissues. Okay, so now we've established the blood stage life cycle of disease pathology. But the beauty of the parasite is that now it has to facilitate its own transmission to the mosquito following the next bite. So certain parasites within that life cycle, within that blood stage life cycle, make a decision. We still don't understand what that decision is, but it's a decision to become sexual. And so they form gametocytes which circulate in the blood, are taken up by a mosquito in the next infectious bite, and then they go into the mosquito midgut. And there they basically they receive cues, which again we don't fully understand, to basically start to mature and for gametes to undergo fusion. So this is male gametocyte maturation, and this is a process called exflagellation. You can't really see, but they basically look like little spermatozoa. They break out of the infected red blood cell. They start to bring in red blood cells around them into these sort of exflagellation centers, these clusters. This is an EM of the same image. These are wonderful microgametes. And then basically you get gamete fusion. And the result of gamete fusion is this motile zygote, which I think is one of the coolest cells I've seen in biology, which is called the uokinete, which basically undergoes this corkscrew motility, and its job is to find the edge of the mosquito midgut. It has to crawl. It's like being put in a dark room and being told, find the edge when you don't know where you're going. But the parasite has evolved an incredible sort of set of motility and strategies to find that epithelial barrier and then break through to establish the next stage of infection. Okay, so the parasite's had sex in the midgut, it's broken through the epithelia, and now it has to form the next stage for reinfection. It has to reform those sporozoites. That uokinete was the only diploid stage in the life cycle. So that having burrowed through the epithelium, the midgut of the mosquito, the parasite then forms these wonderful cysts on the midgut wall. The mosquito actually doesn't sort of suffer too much from this process, which is quite interesting, and forms these incredible cysts which are just bursting, again through remarkable cell division with hundreds and hundreds, if not thousands, of sporozoites per oocyst. These then rupture into the cavity of the mosquito. Basically, they just infest the whole mosquito. Every single tissue of the mosquito is absolutely infested with sporozoites. And as a result of that, the salivary glands, which are actually targeted by the sporozoites, get heavily infected by the sporozoites, ready to reestablish, again with our friend the sporozoite, going back into the mammalian host. So that's kind of pretty much the whole life cycle of the parasite. And I think if you could take one thing from that life cycle, you would take that appreciation that this is a single cell parasite, one genome, and yet throughout its life cycle, it's constantly doing these caterpillar to butterfly transformations. This incredible plasticity within this single cell to keep changing. And in some ways, I feel like that's one of the reasons why I like to work on malaria, because it's just such a wonderful eukaryotic cell to work on for those reasons. So why is malaria disease and the malaria parasite so hard to control? So I'm going to try and sort of cover some areas which contribute to that. So one key area is that it keeps changing its coat, avoiding immune detection, and I'll cover some of these other areas along the way. So one of the incredible achievements of the parasite, you're filled with trillions of red blood cells, so it's a perfect cell to to invade in order to hide yourself from the immune system. But one of the problems is the red cell has gotten rid of all of its sort of translation and expression and secretory capacity. It's not a bag, but it's basically quite an empty cell. So the parasite, once it gets in there, it undergoes this incredible transformation of the infected red blood cell. Alan Cowman has sort of described this as looking a bit like a naked chicken. It's sort of all knobbly, and but basically the parasite has done this incredible transformation of turning what is basically a non-secretory, quite inactive cell into an incredibly active, secreting and feeding cell. So this is a slice through a cartoon of that infected cell. So we have the red blood cell itself, we have a vacuole in which the parasite lives, and then you have the parasite itself. And sort of some appreciation for the complexity of what the parasite had to do in order to achieve this incredible remodeling of the host cell. Another problem that the parasite faces, it's inside a cell, and it's starting to remodel the cell, but it doesn't want to get cleared by the spleen. It doesn't want to get cleared out of the circulation because it needs to undergo its development. So what it does is it makes these incredible sort of macromolecular complexes on the surface, which are called knobs, and these enable it to actually adhere to surface epithelia and stop itself from being carried through the flow system. So if you think back to that in vitro movie I showed you of those parasites sticking to that artificial epithelia, that incredible ability to withstand the flow forces of blood, which are quite incredible. And in this EM, which is really beautiful, you can almost get that sensation of molecular interactions between the parasite, the infected red blood cell, sorry, and the host cell epithelia. Oops. Okay. So the basis of that ability to adhere to surface epithelia is carried out by this protein PFEMP1, which is basically the major virulence factor, because you can imagine that without that protein, the parasite will basically carry on through the bloodstream and the immune system will have a much better chance of clearing it. So the process of getting PFEMP1 onto the surface is incredibly complex. It ends up with this protein bound by this knob-associated complex of proteins and then sticking out. And it's the extracellular domain of that protein which basically allows the parasite to adhere to those epithelial surfaces. However, that raises another problem, that if you're a parasite in a cell and you're doing everything you can to avoid immune detection, if you start putting a protein on your surface, of course, that's the first thing the immune system is going to start seeing, and then you've almost defeated the object. So what the parasite does is that not only does it express, it undergoes this process of antigenic variation where it expresses only one PFEMP1 at a time, but it has 60 from which to choose from within its genome. But not only are there 60 different copies that keep changing, each of those has many modular domains which are constantly under evolutionary pressure to diversify. So you have this incredible divergent diversity-creating mechanism where the parasite can constantly change its coat to avoid immune detection and at the same time achieve that ability to stick to epithelia and stop itself from being cleared, which I think is also an incredible biological process. So then what you get is this process of antigenic switching where the immune system will do a good job at detecting a variant on the surface, but then the parasite will switch. And so that parasite with a new variant will increase in the population. The immune system comes in, will clear it, but then the parasite comes back again with a new variant, and so on and so forth. Remember, it has up to 60 different variants it can choose from, and this incredible ability to change what it's doing to avoid immune detection. We don't understand fully how the parasite's actually able to achieve that antigenic switching mechanism. We know that this is an IFA fish that was done by Ali Marti when she was a PhD student in the infection immunity division. And it just shows here is a DAPI staining for a parasite nucleus within a red blood cell. And what we can see is that there's this artificial label for an active cluster, and when you activate it, which is a complex process which I won't go into, they start to co-localize at the periphery of the nucleus. So what this is suggesting is that the spatial localization of a chromosome end or an active component that you're trying to switch on, where it is in the nucleus determines how it's switched on. And that's how we think the process of antigenic variation is actually achieved, although it's not fully understood. Recent work, which I've had a little bit of involvement with, but also other groups around the world, have started to look at actin, which it looks like actin actually may be a mechanical force able to actually push around those chromosomal ends or something to do with actin to actually activate different VAR genes at different times. And so that's kind of an emerging area of understanding. But I think the bottom line is we don't really fully understand the basis of antigenic variation. A really nice study that just came out from a group in Singapore, from Zibi Bosdech and Alfred Cortez in Spain, they did a really nice study where they took parasite clones, they cloned them out, and then they just did transcriptomics on them. And what you can really see is that this is one parasite line which has made 6 clones, and what you can really start to see, the information there is unimportant other than each individual clone is starting to vary different protein families. So it seems that the parasite has this general bet hedging strategy of constantly just allowing flexible expression of different members of different antigenic families. So not just PFP and P1, but many different surface proteins, whether it's on the blood stage, the free blood stage merozoite, or whether it's on that infected cell. So that ability to constantly avoid immune detection. In addition to being able to actually constantly change its surface coat, the parasite's also under incredible selection in a sort of an evolutionary arms race, both to mutate but also to switch its actual biological strategies. So here we have a slice through this 1 micron size merozoite. This is the stage that establishes blood stage infection, that little one I showed you invading in 30 seconds. It's a eukaryote. It's got a nice nucleus and other subcellular organelles. The way it actually invades a cell, this 30-second process, is also quite complex. It sidles up to a red blood cell, kind of has a sort of a low affinity interaction, and then it recognizes that it's a red blood cell, forms this beautiful structure which we don't fully understand called a tight junction. It then starts to make its own vacuole, squeezes through, releases what we think is an immunological smoke screen of antigens which are shed off the surface, and then establishes infection. And just to reiterate that point, that this is one of those few stages in the life cycle where the parasite's actually exposed to the immune system. So a lot of these surface antigens are really immunogenic and the basis for a lot of vaccine work, which I'm going to talk about in a bit. It's quite a complicated picture, but basically you have this parasite, which is the merozoite recognizing the red blood cell and then choosing to invade the red blood cell. And what we know is that this involves a number of different receptor-ligand interactions, but it doesn't have to use all of the interactions at once. In fact, it can change the different interactions it can use. So you can sort of think about this like the parasite has a number of different keys, and it can get through a number of different doors into the red blood cell. So, for example, a parasite might choose to invade via this glycophorin A dominant route, glycophorin A being one of the dominant receptors on the red blood cell surface. But if you select for parasites in the presence of an inhibitor of glycophorin A, or you cleave off glycophorin A, the parasite just changes into one that can invade independent of glycophorin A and actually invade via the complement receptor 1. Via a different interaction. And this was a paper in Science a few years ago from Alan Kalman's group, where they showed that if you basically knock out or select for parasites in that way, that you actually get a complete change in the biology of the parasite and its ability to actually get in. So under those specific selection pressures, the parasite is actually able to respond to the process of selection. Of course, another means of responding is mutation. We're dealing with a parasite with a very fast generation time, only 48 hours, and a normal eukaryotic potential for mutation. But of course, when we're only reproducing every sort of every 30 years versus a parasite that's reproducing every 48 hours, there's a much greater potential for mutational emergence. So these are just some maps of the emergence of drug resistance. So since we've started using basic anti-malarials, the incidence of anti-malarial resistance is obviously massively rising. A few years ago, artemisinin was seen as the savior of malaria science in the sense that this was a drug rediscovered from Chinese medicine, which was able to be an incredible curative drug, but now already in areas of Southeast Asia, they're reporting resistance to artemisinin. So there's this constant battle against a parasite which is able to not only avoid things like the immune system, avoid strategies that we might throw at it, but also to evolve drug resistance to some of our frontline candidates. And finally, I guess an area I feel least qualified to talk about is immunity to malaria. I believe later on in the lecture series there's going to be a sort of a more intensive talk on immunological responses to infection and malaria. This is a graph that is often shown in malaria talks, and it really just demonstrates the complexity of the immune response. So you've got very, very young children actually receive a lot of maternal antibodies to malaria and actually reasonably protected from malaria. But as soon as they're sort of, what's the word? Weaned, I guess, and have started to sort of take on their own immunological response, they're incredibly susceptible to malaria. And so you get this peak in sort of severe disease for very young children. This is why children are the main group that actually die from malaria. Then you get a sort of a more severe or mild disease, which is quite prevalent in young children as they start to develop their immunological response. And then slowly the incidences of death actually start to decrease, and you get sort of parasitemia and a lifelong potential for infection, and eventually you do sort of accrue this sort of level of immunity to malaria. But as actually we know from personal experience with a friend of the family's my family who grew up in Africa and then as an adult moved to the UK, then went back, got malaria, and actually later on succumbed to the disease, even that immunity to malaria is not lifelong, that your body quickly forgets. And we really don't understand why it's so hard for the human immune system to not only launch an immunological response against malaria, but also to remember the malaria parasite. For some reason, it seems to be a very easy parasite for our immune system to forget. So that's all the kind of doom and gloom. Can we fight back? So certainly, in terms of human history, human populations have evolved some genetic strategies to actually protect us from malaria infection. So there are very sort of well understood mechanisms of resistance, such as sickle cell anemia, which most of you will have heard about in sort of high school. And then I just want to go through a few other aspects. So I've touched on this idea that the human immune system, although it's poor, it actually does develop. So we are able to launch an immune response against malaria. And in some ways, that's the basis of all our strategies to develop an anti-malarial vaccine, is that immunity can arise, so therefore it should be possible to stimulate it in a way which is very specific. So these were absolutely classic experiments in the 1960s, which showed that passive transfer of antibodies from a malarial immune individual to a non-immune individual were actually protective, and later on showing that those antibodies actually could block the ability of the parasite to get in. And in some ways, that's established the whole program of international research to try and develop a malarial vaccine, an anti-malarial vaccine, which could elicit this same type of response where if we had the right antigen or the right cocktail, we'd actually be able to encourage an individual to make an anti-malarial response to protect themselves from malaria. So that's our rationale. But I think it is worth stressing that to date there is not a single human licensed anti-eukaryote vaccine. So there's plenty of vaccines out there which works sort of so-so against viruses and bacteria, but there is no vaccine against a eukaryotic pathogen. There are in pigs actually, but not for human use. So that's kind of part of the challenge that we're really facing. These were the good times when you could do experiments on prisoners in US institutions. This is the prisoner number here, and then he's being fed, or the mosquitoes are being fed off him, and that was where they sort of established some of the basics of if you inject people with malaria, then how long does it take to make an immune response, et cetera, et cetera. So these are some of the papers that came from that time, and it established this idea that if you were able to make an attenuated parasite and then inject that into an individual, they would launch an immune response, and then later on, having been challenged with an infectious mosquito, they wouldn't actually get malaria. And so that was, this was at work. A lot of volunteers and a lot of prisoners kind of gave consent. I think they got reduced sentences if they were helpful. So this kind of established the idea that maybe an irradiated or an attenuated parasite would actually protect. And therefore might be the basis for a malarial vaccine. More recently, there's been an interest in developing, rather than irradiated sporozoites, and actually a genetically attenuated parasite, whether a sporozoite, as this paper was all about, or a blood stage attenuated parasite, where you could inject an individual and they would launch a similar immune response and then be protected against malaria. So it's just our sporozoite movie again. For a genetically attenuated sporozoite. And this is work that's actually been going on here at WeHi in collaboration with a group in Seattle. This is one of my favorite science papers, I think, of all time, because it really tells you how if you get a bad result, you can still publish in Science if you say it right. So this was and I kind of apologize because I shouldn't really punish these guys, but I feel like this is criminal, this paper. So basically, we report that irradiated sporozoites administered to 80 volunteers by needle injection was safe, but not actually immunogenic or protective. And then our data shows that basically administration of vaccine will lead to protection of infection with PF malaria. Yeah, how you get from there to there, I've got no idea. But basically, this was the first trials, which is quite an achievement, and actually that is worth celebrating, of making a live attenuated vaccine and then showing that it doesn't actually work very well, and therefore we need to make it work better. But anyway, so there's still a lot of room out there for any budding young vaccinologist who wants to get in on the game. So I kind of raised these questions about a whole attenuated parasite strategy for a vaccine, which is still being pursued and I still think is very valid to pursue. You know, things like irradiated parasites, can we standardize the dose? Can we actually define a mechanism of delivery? Will a genetically attenuated parasite find a way to break through? I've already shown you lots of evidence that the parasite has a very plastic ability to kind of keep changing its immunological coat. So maybe it'll find a way through. Can we even grow enough sporozoites or those kind of parasite stages to immunize all the children who are at risk? And then how on earth do you get a parasite to areas of rural Africa where there's no running water or electricity and you need to carry the parasite on dry ice and then administer it intravenously? So it's clearly quite a challenge that we face if that's going to be our strategy. A very different strategy is the idea of actually just finding an immunogenic protein or protein component, which will, or even a carbohydrate, which we can conjugate together to form a vaccine that elicits enough of a response that it's as good as being injected with a whole parasite. And that's really the basis of probably the most celebrated of potential malaria vaccines, which is called the RTSS vaccine, which is being pushed by GlaxoSmithKline and a whole suite of international partners. And although it's not been great, the current trials are seeing an approximate protection of about 40% against severe disease. So not against infection, but against severe disease. So this is a recombinant antigen in a formulation which is injected into the arm and then gives you some degree of protection. So that's certainly promising, but 40% is not fantastic. But I guess if you're talking about 1 million children dying a year, 40% of that would be pretty good. So there's definitely hope within the subunit vaccine field and as that pushes forward. But certainly, with just those two examples and many of the other examples, our understanding of parasite biology is clearly still very weak in the sense that having had sort of transfection technology for only, say, the last 20 plus years, we're still very far behind a lot of eukaryote or cancer biology or even HIV biology in terms of really understanding how the parasite works. I guess one of our key tools now is that we have both the genome of the human host, the parasite, and the mosquito vector. So we live in a time where it is actually possible to do incredible host pathogen biology, whether the host is the human host or the mosquito host, and really take a systems biology approach to malaria. And that's really all the rage right now. We just had a malaria meeting down in Lawn, and a huge number of groups are taking these sort of systems biology approach, whether it's transcriptomics, genomics, whole high throughput imaging of parasites. So lots of different sort of systems types of strategies for understanding malaria parasite biology. And as I mentioned, certainly one of the key contributions of WEHI malaria science to the question of malaria parasite biology has been the development of transfection, which was sort of developed by two groups, one at NIH and one here at WEHI. So if you're thinking about, okay, you want to get in on the malaria game and you want to make a contribution, you, I guess, in terms of, say, drug discovery or vaccine discovery to some extent, there are two strategies you could take. One is to focus on one particular aspect of biology. So let's say a part of biology that only happens here, and let that be your focus, whether it's a drug or a vaccine target. Because obviously, if you stop parasite infection here, you stop transmission, so therefore you stop all of life cycle progression. And you could think of any number of ways that you could target that. You could target the process of invasion, you could target the process of remodeling the red cell, sexual development, or transmission. And these are all active areas of research internationally, different groups trying to target those different aspects of biology. I'll just give you one example of a pathway which is actually quite drug targetable, which is also a key research interest here at WEHI. And that's this process of remodeling. So this is a really nice paper done in 2001 from Alan Cowman's group, where basically a very simple strategy. You take a protein which is known to be part of that whole remodeling process, exported to the infected cell, and part of that whole knob formation. And then what you do is you start to make deletion constructs. You see at what point in the deletion construct does the protein stop getting out. So here you can see that 120 amino acids of the protein gets out, but only 60 amino acids don't get out. And this established this idea that there's a motif in this N-terminal region which tells the protein that it has to get outside into the infected cell. So this 60 to 120 amino acid sequence contains a mystery motif which must be involved in that export process. And it actually took quite a while to actually find that motif. But when it was found, it was two groups, Alan Cowman's group and the group of Kasturi Halder, I think at Chicago, is that right? Or Notre Dame. They discovered that there was this pentameric sequence motif, RXLXE, which was the signal, the barcode that told the parasite to take this protein and export it to the outside. And so that's just a five-letter code that is a key determinant of virulence. that all those proteins that need to go out to remodel the red cell to enable all that whole feeding machinery is encoded by that simple barcode. So it's quite a simplistic sort of bioinformatic experiment, but actually took a long time to find, and when it actually came out was quite revolutionary. So here again you have a wild-type pexyl protein. The protein goes out, fills the red blood cell, but when you just mutate one of those residues, the protein can't get out and gets stuck in the ER. This led to a second stage in the discovery process, which was actually discovering a protein that actually cleaves this pexyl, this 5 amino acid motif, and that's an ER-resident protease called plasmepsin 5, that once it's processed the pexyl motif, the protein then gets exported out to the red blood cell. And then a final stage in the process was the identification of what we think, we still don't know, is the complex that sits in the parasite or in the parasite vacuole, which actually enables the parasite to export these proteins into the red blood cell itself, the proteins that need to get out in order to start remodeling the red blood cell. And just sort of my own my group's small contribution has been, recently we've started doing super resolution imaging of some of these proteins and starting to look at the macromolecular structure of this complex of proteins which we think may be the transcon, although it's not been proven yet. So you can think of that as, you know, we're thinking about drug targets. How might we make a new drug against malaria? Let's say we go for the blood stage. We could go for any one of these processes. We could go for pexyl processing, target the protease that does that processing in order to stop export of those key virulence factors. That would clearly screw with the parasite, so the parasite wouldn't develop, and so on and so forth. That'd be quite a good treatment drug. Similarly, we could go after protein export itself. We could try and jam the translocon, stop proteins from coming out. Another strategy you could think about taking would be to go for something a little bit more generic. Is there a part of the parasite life cycle which is common both to, say, invasion, but maybe the mosquito stage, maybe colonization of the midgut, and maybe infection, whether it's of the salivary glands or the liver stage. And this is actually my own pet interest, which is how parasites move. Because movement, as hopefully you will have got an appreciation for, is key throughout the life cycle, whether it's infection of the red blood cell, carriage through the epithelia of the midgut to actually establish those cysts on the midgut, or again, infection of the salivary glands or parasites. So the parasites are constantly on the move. What's also really nice is that eukaryotic cells have many different ways they like to move. You've got cells like your immune cells, or this keratinocyte, this is an acanthamoeba, which move in this sort of amoeboid-like movement, crawling, pushing out fronds and fronts, exploring space. That's a very classic way of that eukaryotic cells achieve motility. An alternative way might be to use a flagellum. So this is a leishmania parasite, or it could be a spermatozoa. And we know that the parasite can actually form a flagellum because the microgamete has it. But the malaria parasite is not using this strategy either, and it's also not using a common virus or bacterial strategy where you modulate the host cell actin to actually propel yourself around. These are actin comets. The actual virus or bacteria is right at the head. So these are beautiful ways that cells use to move, but what's nice is the malaria parasite doesn't use any of them. In fact, it uses its own unique method of moving. And how does it move? It glides. That's the best slide of the whole talk. Okay, so malaria parasites glide. They use this completely unique means of traversing substrates and invading cells. And we don't fully understand it. And certainly my own interest in it started when we started to look at whether the proteins involved with gliding were conserved in the blood stage. And it turns out that whether you're talking about a sporozoite, a nookinete, or a blood stage parasite, despite looking radically different, and I remind you the caterpillar to butterfly transformation, they're using the same basic molecular process to actually move. So this is our best bet about how we think parasites move. It's a cartoon. It's not reality. So here we have the inside of the parasite. Here we have the outside of the parasite. We have a myosin motor. So this is the force for motility. This gets recruited to the inside of the parasite to a sort of an anchored membrane complex. Then the parasite, based on whether it hits a substrate or a cell, it secretes surfaces onto the surface adhesins which allow it to stick to the substrate, and then some signal comes in to actually polymerize the filament of actin, and then you get this connection. So you can think about this like a rowboat down the river. You've got your rower and you've got its oar, and then that's associated with the substrate, and then this happening as you zoom out, little Pac-Mans, little protease, As you zoom out, this enables the parasite to move across the substrate. So this is how we think malaria parasites move, and indeed the phylum to which malaria parasites belong, called Apicomplexa. That's how we think it moves. So immediately that might fire off ideas in your head about how one might design a drug or a targeting strategy that would affect the ability of this motor to function, and that would clearly be profound. If you had a drug which was very stable in the bloodstream, was taken up in a mosquito feed, that same drug that would stop infection would also stop the ability of the parasite to colonize the mosquito, a transmission-blocking drug, which is an idea that people are thinking about. So you could actually try to disrupt actin itself, which is one of my own interests. You could try and target myosin, the motor. You could target the regulation of the motor. There's very complex signaling pathways, like many things that I've talked about, but certainly here there's a key phosphorylation cascade that we don't fully understand, which is involved in signaling the motor to be active. You could target that. There's any number of different kinases one could target there. You could also target proteases, which are important for releasing those adhesion plaques. There's a very nice set of data coming out now from a group that has shown that if you mutate the protease cleavage site, the parasite tries to move but gets stuck. It can't unstick itself to keep moving forwards. So there's clearly proteases that one could target there, and any number of different parts of the motor function. There's parasite biology, which you could target, but another very prominent and in some ways very straightforward way of trying to control global malaria is to actually target the vector itself, start targeting the mosquito. So here is a photo of a factory. I'm not actually sure where it is, but it's from the Rollback Malaria campaign, which is a partnership with a number of different big players, Gates, WHO, Rotary International, and they're basically trying to supply everybody who needs one with an insecticide-treated bed net, a very powerful way of stopping transmission of the disease, if you can stop being bitten from mosquitoes. One of the challenges that arises as a result of that is our whole immunological response to the parasite. If we stop getting exposed, would that be a disaster waiting to happen as those insecticide bed nets, treated bed nets start getting holes in them, etc. But certainly there is that strategy, and there are definitely signs in certain communities that that's really starting to pay off, as communities which have been netted completely are starting to see rapid falls in the number of malarial incidences, but I guess time will tell. There are other strategies that Gates is starting to sponsor. This is one of my pet favorites, which is a laser-guided mosquito killer which sits on the front of a helicopter, and that basically somehow this manages to kill thousands of mosquitoes per minute as it flies through the jungle killing mosquitoes. I can't believe they funded that and not my idea. It's okay. I'll get over it. Perhaps a more sensible suggestion is work now being done at Monash University. It was originally started up in Queensland by Scott O'Neill's group. He's now the Dean of Life Sciences, I think, at Monash. And his group have found this bacterial symbiont which lives within mosquitoes, which in Aedes aegypti, which doesn't carry human malaria, protects them from transmitting dengue fever. And there's now trials which have been reported in Nature where they've shown that this mosquito is able to actually establish itself quite successfully in the population. And because its resistance to dengue, it would actually lead to a dramatic reduction in dengue fever locally. One of the side things they noticed in their paper, you'll notice it just says plasmodium. The plasmodium that this Aedes is actually resistant to is chicken malaria. So this is great if you're a chicken. But so far, attempts to actually establish Wolbachia symbiosis in Anopheles mosquitoes, which are the natural vector of human malaria, have been unsuccessful. But you can imagine if that does work, and I would imagine it's only time before they get it to work, how powerful it would be to release this Wolbachia-infected Anopheles population into the wild to actually just stop transmission. That would be a very powerful way. Another strategy that other people are using is actually to release genetically modified mosquitoes, which either can't have sex or maybe don't don't carry the infection for some reason. So another strategy. So I've sort of tried to introduce the idea of targeting an individual part of the life cycle, the broader life cycle, or indeed target the vector itself. Okay, so we're getting near the end now. So what I guess I've tried to show you, so there's a number of reasons why malaria is still out of control and is still outwitting our best efforts to control it. But we certainly are in an incredible opportunity in this I reckon in the next 25-year period. We have a number of tools that have never really coincided before. We certainly have at least a 40% efficacious vaccine against severe malaria, and I think that's only likely to improve as formulations and different antigens and different ways of producing the antigen are covered. I think for the military and for tourists, there is really the possibility that there will be a genetically or a irradiated vaccine, an attenuated vaccine that you'll be able to get from your GP, which would actually protect you. And that would be quite an achievement. I don't think that's actually, my personal views, I don't think that's going to mean much for the developing world. But certainly there are a number of vaccines now in the pipeline. The rollout of bed nets is clearly going to be a very significant player in transmission of the parasite. Resistant vectors, as I talked about with Wolbachia, new insecticides, which I know very little about, but I know they're being talked about. And then, of course, new drugs, which is an area I'm particularly interested in, this idea of identifying new drug targets and putting them together in different combinations to try and beat, just like you would in bacteria, beat the emergence of drug resistance. And an area I haven't really talked about, but is this idea of improved diagnostics. One of the key challenges, I think, and I don't say this just as lip service, but I actually believe this is really important, that our understanding of the epidemiology of the parasite and diagnostics of the parasite is critical, not only now, but looking to the future. If we do start to control malaria, one of our biggest challenges is to stop it coming back. There are very well-characterized historical stories, for example, in Sri Lanka, where malaria was all but eradicated, and then they released their sort of control program because it had gone, and then when it came back with 10 times the vengeance that it had the first time round. So it's not just our control of malaria, it's actually being able to control it and then monitor it long term. And there are parts of the life cycle which I haven't mentioned where the parasite's actually dormant for some species in the liver, and we need to have biomarkers for that dormancy, otherwise we'll never get rid of malaria. So clearly there are very important areas of diagnostics and understanding the disease epidemiology which we need to do. And of course, we've got other problems in the game, things like poverty. This is really a disease of poverty. If you cleaned up the whole of Africa and gave them all first world hospitals and bus stops, that's one of my favorite statistics, is your greatest chance of dying from malaria is how far you live from a bus stop. So in some ways, it's got nothing to do with disease sometimes. It's often to do with primary healthcare and the provision of infrastructure. So, and I'm kind of saying that apologetically because I'm a cell biologist who likes to work on the parasite, and but I think economics has a key role to play in the control of malaria. War, I mean, some of these areas are some of the most unstable areas in the world, so it's going to be quite difficult to eradicate a disease or control a disease in areas where you kind of get people to talk to each other. And also, just to reiterate that idea that we're dealing with an ancient foe, which has got an incredibly complex biology, constantly changing, and is very well adapted to rising to any challenge that we throw at it. And then my favorite quote: malaria is about to be totally eradicated and you will never make a career, let alone a living from it, which was advice given to Robert Desowitz in the 1950s in the middle of the control and eradication program. And I think you'll find that in Melbourne alone, there's probably more malaria researchers per capita than any other city in the world. So there's clearly a huge amount of research that we need to do to understand this quite remarkable and talented pathogen. Thank you.
Thanks very much, Jake, for a really quite magnificent overview of malaria and demonstrating its complexity, which really now enlightens me as to why it's so hard to tackle that bug. Questions? I can't see you, but up back.
Hi, Jake. It's Janine.
Oh, hi, Janine.
That was a beautiful overview.
Thank you.
My question relates to the turning off of PFMP1 expression, which would predict to allow for splenic clearance as in the carb knockout. But I was just wondering if any progress has been made on more recent insights for possibilities for intervening with PFMP1 expression.
Well, I guess there's been a couple of strategies, as you know. One is to try and make a sort of an attenuated, genetically kind of modified parasite that would express all of its PFMP1, either on the surface or inside, to kind of really prime the immune system for everything it might possibly see. I'm not sure if that's progressed so much further. I think with a lot of these ideas, it's actually making the parasite under GMP and delivering that kind of potential. Certainly, in terms of drugs to actually perturb export, I know that there's a very active program in Alan's Cameron and Justin Body's group to do that very thing, whether it's targeting the translocation or whether it's targeting some other process in the maturation of a protein as it goes through. I have no idea how far they are along in that process, but I would assume you have to just watch this space. Any other questions? Lynn?
You reminded me of the more benign malaria species that we don't hear about very often. In the context of thinking about attenuated strains. Are Plasmodium ovale, for instance, or the other that you mentioned, any use in informing or even being attenuated versions of the parasite?
Yeah, I mean, I think now, I'll answer a slightly different question, which is that there is definitely an increased perception of the importance of understanding the other malarias. You know, falciparum has dominated the game for a while, and rightly so, because it's the cause of most death from malaria. There is now an appreciation that we really need to understand the other parasites. Vivax is proving very difficult to work with, but it's clearly one of the keys to unlocking or controlling disease from malaria. And ovale could actually be a very clever model for Vivax. It's not nearly as lethal, but from my understanding, it also does form this dormant stage in the liver. So certainly, we need to understand the whole biology of all of the plasmodia for the different insights they gave. And in some ways, they give us insights into biology which are kind of comparable. So for example, Vivax doesn't sequester in the same way that falciparum does. So how does it avoid splenic clearance? So one of the ways it gets through is it's much more flexible cell. So it doesn't actually clog up, it actually comes through the spleen. So different parasites are taking different strategies to avoid the same basic problem. And I think that kind of highlights quite nicely how, you know, we're talking about, they're only distantly related. We call them malaria, but they're actually probably more ancient than we are to, say, a fruit fly. Within that group, I think there are some compare and contrast aspects of biology. And just to sort of extend that one more, is that Knoll's eye, it only has a 24-hour life cycle. When it does come into human populations, it's quite a nasty pathogen. But it was seemingly moved by the same way as falciparum does. So actually, it's a bigger cell. It's kind of it's in some ways easier to work with. It grows much faster. So it could be an incredible model for understanding motility in falciparum if we were able to work with Knoll's eye red blood cell infectious stages. So that doesn't really answer your question, but I guess that the Plasmodia as a group can certainly teach each lessons to different species. Yeah, questions?
With the liver stage of the parasite, do you know if there's a lot of remodeling of the parasite when the sporozoite enters, analogous to what happens in the red cell when the merozoite invades?
Yeah, people are just now working on it. I mean, it's just started now. One of the problems is, I don't know how many work with hepatocytes or with liver cells. They're quite difficult to work with, and we don't really have a good model, for... well, first of all, we can't make in vitro human falciparum sporozoites to do the infection studies. So you need an insectary, which we now have at WEHI, which is really exciting. Hopefully by the end of the year, we'll be able to actually look at the insect stage of biology, which is really very exciting. So in order to get a hepatocyte infection, you need a live mosquito. So let's say you've got that and you establish an infection. Even that only seems to work in one cell line at the moment, which is the hep G2 cell line. It would work in primary hepatocytes, but they're very difficult to work with. And then the early suggestions is that there is probably a remodeling process. You would expect it. I mean, you'd sort of say it for sure exists. But now we've just started to look at it, and groups are starting to find that there is evidence that proteins are exported into the hepatocyte. Certainly, it has to suppress things like apoptosis. I put that out there just for all the people who work on apoptosis in the institute. The parasite's clearly got means of stopping the host cell from dying, and in other stages of actually initiating cell death. So, very new area of study.
What about the step from getting to the mosquitoes to the liver? Is that targeted at all, and how successful is it at that step?
It seems to be based on the levels of sulfation on surface glycoproteins. And I believe one of my students said that if you add enough free heparin sulfate to any cell, it will basically invade that cell. So I don't know what it is about the liver cell, and that's out of my area of expertise, but I believe that the liver cells in particular are very well sulfated, and that's kind of one of the signals that the parasite gets, that it knows it's in the right place. But there's probably a lot of complex receptor-ligand interactions in terms of the parasite. I think that's another kind of beautiful aspect of the parasite biology, is that it needs to know where it is all the time. It has to have, the all singing and dancing GPS systems say, I'm at a red blood cell. This is the part of the biology that I'm now doing. I am in a mosquito midgut. This is the part of the life cycle I should be doing. And so how it gets from the skin to actually invade a hepatocyte is assumed to be quite straightforward, but I imagine it's incredibly complex. Or maybe it's very simple. Who knows?
I have a very naive question.
Respond Met Call Ward 3 South, bed 12. Respond Met Call Ward 3 South, bed 12.
You took a parasite from the blood phase and stuck that particular parasite into a mosquito. Would it live? Would it change its whole biology and become the butterfly rather than the caterpillar?
That is a good question. I think it just gets digested, unfortunately. The parasite that does survive in there, it seems to use those cues specifically for its biology, and it must have means of protecting itself. So we think actually there's a surface protein which forms these wonderful triangular structures. A lot of the structural biology actually of malaria parasites are really interesting. They form these completely wild kind of protein structures which are not found anywhere else. So this is a triangular structure which seems to coat the surface of that gamete, and that actually seems to protect it from stomach digestion, and may also be part of the signal-mediated process to tell it to activate. So the answer is no, but I think there are other aspects of the life cycle where that question might be interesting. You know, if you took the wrong stage and put it somewhere else, what would happen? But I think generally it's a very well-ordered cascade that it knows where it is because it can't switch places in the cycle. But
if you targeted tricking it to think it's somewhere else, then it might die?
That's a very clever idea. I mean, in some ways, I think some people are starting to look at that in terms of they're creating parasite lines to try and tell the viewer when they are committing to the sexual stage. And we don't know the cues for that. But I think as we understand the genetic switches, the transcription factors that regulate that process, you could well imagine that you could mess with that system and either block transmission or maybe get the parasite to commit completely to gametocyte biology and therefore stop its progression. But then that will make it very infectious. So trade-off. Maybe, okay, one more question.
Jay, thank you for a very fascinating seminar that you gave. So throughout the whole seminar, you've highlighted a lot of the major approaches that malaria researchers are doing to eradicate malaria. So do you think, which of all those approaches that you think is our best bet in eliminating malaria in our generation?
Stop poverty. Yeah, I think that's, the honest truth is, like most infectious disease, sort out the infrastructure in Africa. If you can do that, then I think you'll cure malaria. I don't know if you'll eradicate it, but in terms of parasite biology, I think the blood stage in terms of disease is the best thing to work on. If you want to stop actual establishment of infection, you can't really work on blood stage. You've missed the game. And I think transmission blocking, purely because it's such a radical thing to do, to sort of think about, so there are programs now where they think about immunizing a whole population to stop transmission. So if you get that immunization, your chance of getting malaria is exactly the same as it was before you got that immunization. But it's a kind of a group, a sort of a herd strategy that you're actually protecting the village from transmission. And I think there's a really cool idea in there. So I think if you can, basically, I guess the most obvious way, and this is the way that everybody's thinking about it now, is that you combine all of those strategies in one wonder drug or one wonder vaccine. You target all three stages of the life cycle, liver, blood, and transmission, in one vaccine, and then you know everybody gets everything for that one vaccine.
But do you think the vaccine strategy will work? Clearly, the RTSS vaccine is not very successful at the moment.
I think vaccination will work radically, but it won't eradicate malaria. So if you want to eradicate malaria, you need the other strategies as well to control outbreaks, to control epidemics. You need all that epidemiology, all that diagnostics. So I think this is why people in the community are so afraid of the word eradication. It's very easy to talk about control, but as soon as you start talking about eradication, it's a completely different game plan. But we can talk about that over lunch. Maybe stop there.
Yeah, before we thank Jake again, can I remind all the students that there's lunch afterwards and you can ask Jake some questions. I'm sure he'll be very happy to answer. So thanks again very much, Jake.
Sure, man, take it back.
Yeah.